Okay, this next presentation is a PowerPoint on seed propagation. So we're going to start into the production area. Where does it start? Well, a lot of people see it starting at the seed, planting the seed or the transplants or whatever you're starting with. I'm growing strawberries, I get little tiny transplants to stick in the ground rather than seeds. You got to start somewhere. And you could back it up. There's research and other things that go into the process of getting a grower, a farmer, to start growing something. You've got to do all the land prep and there's a lot of business activities that must happen before you get started. But from a production standpoint, we could think of it as starting from the seed. I think that's a good place to begin. So, where do we get our seed? Most of the seed that we use here in the Salinas Valley comes from the western states like California, Oregon, Washington, and Idaho. They are big seed production states, and the western United States has some advantages. They generally have low humidity, which means less fungal and bacterial diseases to deal with in the seed production. Generally mild temperatures, which are good for flowering and good for seed set, and even distribution of rainfall throughout the year. So it's predictable rainfall. And if anything, we get surprised by less rain, like now, and drought rather than unexpected excess rains, especially during the seed set portion of the year, during the summer and possibly early fall months. And so that really provides a good environment. Now, some people might look, Oregon and Washington, they're all about rain. Well, yeah, along the coast, they're all about rain, but two-thirds of Oregon and two-thirds of Washington are dry, high deserts. And so there's a lot of seed production in the desert regions, and some things like spinach, for example, is produced in the valleys of the more temperate rainfall areas of Washington. Another advantage is being isolated from major production areas. So you don't have the pollination problems with pollen crossing with the pollen where you're trying to grow the seed. So the production areas aren't contaminating the seed production or disease issues. So if you do a lot of production, you're likely to have more fungal spores spreading around in an area. And so the areas being separate is really good. And you might think, well, California, I'm, you know, growing lettuce in California. Do we grow lettuce seed in California too? Yeah, but they're different areas. I'm growing lettuce here in Salinas Valley all the way down to the coastal regions near Oxnard or something like that. And I've got some production in the Huron area, a little tiny window in the spring and the fall between the transitions. And we'll talk about those transitions later in the class. We grow here in the summer months. We grow down in Yuma, Arizona. I'm talking lettuce now. Yuma, Arizona is producing for a few months in winter, and then it comes back. But Huron is this tiny little blip in between. And that's it. We regrow the Central Coast all the way down, and we grow a little bit in the Huron area. But there's a whole bunch of that Central Valley where you're not growing lettuce for production. So we can go a little south of Huron, be away from those areas, and grow our seed down past Firebar or something like that. Now, hybrid seed production. Now, that's something that you may have heard of, maybe you haven't heard of, but hybrid seed is seed that is genetically it's genetically uniform, one seed from another, but internally, its chromosomes aren't the same. It's one chromosome from an inbred mother and one chromosome from an inbred father, and those chromosomes are different. And that produces a an advantage in several different ways. But hybrid seed requires some kind of pollination. In some situations, like tomatoes, that pollination might even be done by hand, or it might be done by wind. Corn, for example, is wind pollinated. So we just need to isolate our corn in a way to make sure that only the pollen that is from the correct male is going to get on my female to grow the seed. But if it's tomatoes, I'm sending that stuff to certain developing countries, Southeast Asia, perhaps South America, perhaps, where the labor costs are so low that you can actually get seed that's affordable. But it's very, very labor intensive to produce seed. And so the process has to be managed very, very carefully in order to grow the plants, make the proper pollinations, mature the seed, harvest the seed, clean the seed, store it, package it, whatever you need to do. Now, I do want to go back very briefly to this concept of these pollinations. And I'm just going to show you something here with paint as my whiteboard. So I'm going to create a blank image. And let's say you have a plant. And actually, it's not doing it. There we go. Let's say I have a plant for one gene. It is capital R, little r, meaning it is heterozygous, or it has different alleles, different types of genes, alleles. Allele just means a type of gene for this one pair, because from its mom and dad, it got different things. So I have big R, little r. And so what do? With big R, little r, then my children could possibly be any number of different things. I could be big R, big R. I could be big R, little r. I can be little r, big R. Or I can be little r, little r. There's 4 possibilities of children from this single plant. And that's not good when I'm making seed. So what I need to do is create a mother plant and a father plant that is going to give me what I want if I'm making hybrid seed. If I want it to be big R, little r, then what I need to do is make sure that my mother or father is big R, big R. And then what can it make? It can only make big R, if this is the mother, big R eggs. That's all it can make, because that's all it's got. And I cross that with a male, which is little r, the same gene, just a different version. What can it make? It can only make little r sperm. That's all it can make. So every single child from this is going to be big R, little r. This is now my hybrid seed. And this is what I'm trying to get, perhaps. And this is just one gene that we're looking at. This is repeated thousands and thousands of times along the chromosomes of the plants I'm working with to make seed. But to make hybrid seed, the idea is to get a highly inbred mother and a highly inbred father, so that they are uniform. And if I have a uniform mother and a uniform father, not just for this one gene, but for all of the thousands and thousands of genes in the chromosomes in the genotype of that plant, I can make uniform, what's called the F1 hybrid. The F1, I'm not going to put this on a test, but it means first filial generation. And I cannot write this way, but there's my F1 hybrid seed. And if I plant it right, I can do the work where the mother has what I need from it, father has what I need from it, and I cross it and I make the ideal hybrid. Now there's another type of seed, and we call that type of seed selfed or open pollinated. I'll just put OP here because I can't write very well. OP stands for open pollinated, OP seed. And this OP seed, if I want my children to have the big R gene, mother has to be big R, and then I self the mother plant. I don't cross it with a different plant. I do what's called self-pollinating, and all my children are going to be genetic clones of mother because that's all mother has. She only has big R. And maybe, a little y, whatever that stands for. That's just another gene. There's thousands and thousands and thousands of genes. And the version of those genes, the type of that gene, is called an allele. So for example, every single one of us has basically the same genes with very, very, very minor differences. But we have different versions, different alleles. So we all have eye color genes. We all have the same eye color genes, but we have different alleles, slight variations in our eye color genes that might give us blue eyes, green eyes, hazel eyes, brown eyes, whatever. So those are just different versions. We talk about people having different genes, but that's not really accurate. We have different alleles. So a pea plant, peas can have white or pink flowers, for example. So a pink flowered pea plant has the pink allele. The white flower has the white allele. And it gets a little more complicated than that. We won't get into it. Bottom line is you are making hybrid seed or maybe open pollinated seed, which is easier to make. So here's a nice aerial photo of a flower company, Goldsmith Seeds. Not sure which facility this is, but I remember being fortunate enough to fly into Amsterdam in the Netherlands during the season that they're in bloom. And do you know what they grow in Holland, the Netherlands? Tulips. And to see miles and miles from an airplane coming into the airport, tulips of all different colors and these beautiful patterns, it was an amazing sight to see. So what do you have to do? You have to do things like here is the male portion of a flower, the pollen producing anthers. And so somebody has to come in there and remove those pollen producing anthers and either discard them if this is going to be used as the female plant for seed, or use them to collect the pollen to put on another flower if this is going to be the male plant. So you have to do all kinds of hand labor in many cases, or maybe you'll do labor out in the field. So you need to know the biology. How does your plant actually work? How can you make crosses? dealing with seed production in tomatoes is very different than lettuce, which is also very different than in spinach, which again is very different than in stock or other horticultural flowers, tulips. They each have their own techniques that you need to master. Here's a picture of seed drying. So they're harvesting the plants, laying them out to dry, and then they will be shaken in some way to separate the seeds from the stems, and then go through a cleaner process to remove the rest. Here's some tomatoes, research tomatoes. This is a little different than I did it. I worked in tomato breeding for many years, and I would have paper bags, actually pre-labeled paper bags that we would lay out with stakes to hold them in place. Um, and we would collect the tomatoes, bring them back, and do a miniature version of these sluices. These would be seed extractors. Now, this is a commercial size seed extractor. You would grind up the tomatoes, let them sit for a day so that they partially start to decompose. The acids, the seed separates from the flesh. And then after a day of starting to ferment just a very little bit, you run them through this sluice and you can separate the seed from the other junk. Here's the bottom end of that sluice. Here's a dryer, a very, very large dryer on the outside, and on the inside, there's dry, warm air blown through this as the seeds are tumbling to dry them. I've taken seeds and just put them in a bag, a very tight knit sack, something that might have been the consistency of a handkerchief, but made into a very tightly closing bag. Put the seed in there, throw it into just a regular dryer, clothes dryer, with the heat turned off. I converted a number of dryers and refrigerators and other things for agricultural use in research. And just a regular dryer worked fine. Just tumbled the seeds for a while. I removed the fins on the inside of the dryer so it wouldn't bang around as much as you want your clothes to do it. Perfect. Made wonderful, wonderful seed dryers. Then you have to coat the seed. This you would never see in a commercial environment. These are different types of seed coating. The blue, red, and white probably would indicate different types of fungicides that were used to coat it, depending on what the customer needed. But it could mean other things as well. The company can make the color coding for whatever they want. But in my situation, the seeds were always color coded based on the fungicide used in processing those seeds. This is a separation table. It's slightly slanted in two directions, and then it vibrates. And as it vibrates, the seed move down and to the side, and it separates them out by size. So the bigger seeds are going to move differently than the smaller, lighter seeds. And so you can separate the seeds out into the large ones and small ones on these. There are air blowers and all kinds of different devices used to clean and separate and prepare seeds. Then you put them in some sort of storage, either cans or bags of some sort, to be stored in whatever conditions you need. They can be super deep freeze, or it can be just a modest warehouse, whatever you need. So at that point, before I get into the actual storage, I'm going to take a short break.